Welcome back. What we're going to do now is something that I think is always fun in terms of kicking off the course, which is an exam in biological numeracy. So, just as by way of background to the exam, quote unquote exam, I guess I wanted to just call your attention to two books, both of which I learned about from Bill Gates. And you can decide whether or not you think that's foolish or not interesting, or it may be very interesting, whatever. But here's my take. So the one on the left, Epic Measures, is the story of Chris Murray, somebody who's, in my opinion, maybe his stock has gone down a little bit because of the predictions of his institute about the coronavirus. But at any rate, I'm deeply impressed with this guy, and this is the story of his attempt to try to take stock of the health of everyone on planet Earth. So I highly recommend it. And the one on the right is called Factfulness, and it's by Hans Rosling, who's no longer with us. But you can see from the subtitle, 10 reasons we're wrong about the world and why things are better than you think. And so the Factfulness book is associated with an exam, which is called the Gapminder exam. And frankly, I would be very happy if you would actually go and take this test. As soon as you watch this video, maybe even pause it right now. It'll take you 5 minutes. Just take the exam and see how well you do. And I point out here, showing my buddy Jennifer Lippincott-Schwartz on the bottom, just to say that chimps actually often do better than humans on this exam. So it asks questions like how many of the world's 1-year-old children today have been vaccinated against some disease, etc. So take a look at that. So Mark Kirshner at Harvard made this remark that Ron Milo, I think, passed along to me, and I really like it a lot. Trying to understand molecular biology without numbers is like studying history without knowing geography. And every time I hear that quote, I think about Hannibal and his crossing of the Pyrenees, if I remember correctly. So it's the notion of where are the mountains and where are the rivers and stuff is really relevant. And in the world of biology, even the geography associated with the Wallace Line, the Straits of Lombok, for example, between Bali and Lombok, it's a key thing having to do with the nature of the history of the world that depends upon the geography of the deep straits between Lombok and Bali. Anyway, so our biological Gapminder is going to be this little exam. I show you here Yunon Baran and Ron Milo, with whom we wrote this paper on the biomass of the Earth, which in some ways inspired this exam. And this is strictly for your own fun. What I would hope is that as I go through it, with each one of my questions, that you'll actually stop and you'll write down your own answer before looking at the next part. So we begin. So question number 1 is, what length book would DNA polymerase, including its proofreading properties, copy without an error? So choice number 1 is a book the length of the Bible. Choice number 2 is a book the length of the Encyclopedia Britannica, which is basically one full shelf. And then the third example is the entirety of the English Wikipedia. And so the answer here is English Wikipedia, so 10 to the 10th letters. So you can reason out whether or not you think that English Wikipedia has that many letters, but the error rate in the copying of genomes is order 1 in 10 to the 10th. That is to say that 1 out of every 10 to the 10th base pairs, or every 10 billion base pairs, is copied mistakenly. Given that that I'm recording these lectures as a result of the coronavirus pandemic, I think it's interesting to note that the SARS-CoV-2 virus has a 30 kb single-stranded RNA genome, and it has its own proofreading capacity in order to keep the error rate down, which is a very interesting topic that I would like to touch on further. So this is just to try to give you a sense of this biological fidelity, and this is inspired by this wonderful article by Tanya Baker. And so what she points out is that if we think of the DNA copying apparatus as being the size of a FedEx truck, then the picture would be that it's traveling at something like 500 kilometers per hour. It makes a delivery on both sides of the street every 10 centimeters. It makes its complete daily journey in 40 minutes, and it makes a mistake. It delivers a wrong package only once every 3 years. So that's the nature of the copying. And here I'll just show you a little video.
Using computer animation based on molecular research,
narrating.
we are able to picture how DNA is replicated in living cells. You are looking at an assembly line of miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called a helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time.
So, no hyperbole in there. This is truly a remarkable feat. And, as Julie points out, there's funny business because of the 5' to 3' copying aspects, that the other strand has to be copied backwards, and et cetera. But my main point is, that this fidelity is really something deeply impressive. And here, I'll just give you a little bit of a sense of the data, and we'll come back to this later because one of the themes of the course is life as defiance. Life is defiance in the sense that many, many processes in living organisms take place in ways that I don't know if I'm saying the right word, that basically contradict or defy the laws of equilibrium physics. So here we could imagine that the copying of the mRNA into protein took place. Let's say we only had 2 species of amino acid, let's call them hydrophobic and polar, then we could compute the equilibrium probability of the wrong codon, anticodon binding, and that would give us an error rate. And as I will say later in the course, now is not the time, but we can compute that using the laws of equilibrium physics, which come from Boltzmann, the very same person that so inspired Schrödinger, if you'll remember from that earlier lecture. And we would get an error rate of 1 in 100 rather than 1 in 10 billion. And so there's some funny business going on there. And that kind of estimate inspired people like John Hopfield, shown in the upper right, and Jacques Nignot, shown in the lower left, to in the early 70s posit this idea of kinetic proofreading. And I'm gonna be very excited to talk about that further. So that's the answer to our first question. How many sugar transporters are needed to make a bacterium? So if you think about the miracle of forming a new cell, you basically take a single cell, you put it in 5 ml of clear liquid that has some carbon in it in the form of a sugar, and then it has some salts to provide nitrogen and phosphorus and a few things like that. And it has to actually, in order to make new cells, you have to bring in carbons. And so I'm not going to go through this calculation right now, but as you can see on the left, we start out with this clear liquid, and 12 hours, 24 hours later, you have a completely opaque solution with 5 times 10 to the 9th cells in that material. And we can figure out that or just trust me on this for the moment, because we're going to come back to it. But it takes something on the order of several billion carbons per cell. And if the division time is of order 1,000 seconds, that means 10 to the 6th sugars need to be brought on board every second. And so we'll be interested in this as the course proceeds, and I'm gonna come back to this in the next vignette. So I basically am passing it by right now. This one is amusing. Which is bigger, protein or the mRNA that codes for it? And so I think the reason I find this at least a little bit interesting is that we kind of think of mRNA as being the template or the blueprint for the protein, and so conventionally in our everyday world, we think of the blueprint as being much smaller than the object being blueprinted. But in the context of mRNAs and proteins, there's several ways to see this, but the simplest is probably that, for every single amino acid, you have 3 nucleotides. And the typical mass of an amino acid is around 100 daltons, a dalton being the mass of a hydrogen atom. And the typical mass of a nucleotide is more like 350 daltons, and so you have 3 of those nucleotides for every single amino acid. So the answer is that mRNAs are bigger than the proteins that they code for. So okay, who's more powerful on a per kilogram basis, me or the sun, or you or the sun, and by what factor? And this one's kind of fun and interesting, and this I made reference to when I talked about order of magnitude thinking. I told you that in principle, if you were on the desert island with Wilson, you could figure this out for yourself without really knowing much. I mean, in other words, you could figure out the mass of the sun, and you could figure out the power of the sun, because you might know that roughly 1,000 watts per square meter is incident on the Earth. And so we can use that in a sphere with a radius of 150 million kilometers. That'll give us the power of the sun. And similarly for me or you, we basically put out about 100 watts. I'm about 100 kilograms. That means 1 watt per kilogram. And for the sun, it's actually about a factor of 10,000 less. So humans are way more powerful on a per kilogram basis than is the sun. I think I'm going to skip this one, even though there's some interesting things there. Maybe I won't. So how long to exhaust the ATP metabolite pool in the absence of ATP synthase? So you all know probably that ATP is the energy currency of living matter. And the question I'm asking here is, if you were to turn off the little engines that create the ATP, how long would the ATP reservoir pool last? And so, we can think this through. You're going to do this on one of the early homeworks. So I ask you, if you have 1 molecule per E. coli cell, what's that concentration correspond to? And it's about 1 nanomolar. And so if you look over here on the right, you can see that ATP has a concentration of around 10 millimolar, which means there's on the order of 10 to the 7th ATP molecules in the reservoir of the cell. And we're consuming them at a rate of about 10 to the 6th per second. So on the order of 10 seconds, and they would all be consumed. I wanted to say that we're going to spend some time on proteomics and how well we know the molecular census of living organisms. We'll start out thinking about E. coli, but I'll also pass along to you some ideas from yeast. And the thing I wanted to say here is this is a report of 2 different methods of measuring the subunit counts of the various molecular pieces that make up the ATP synthase. And the thing I guess I want you to appreciate is that on the basis of structural biology, as shown on the right-hand side, that column tells us that, over here, this column on the right, tells us the expected stoichiometric ratios of the different subunits. Then the ribosomal profiling and the mass spectrometry are used to make these molecular counts. I'm going to talk about this later, so this is really just to whet your appetite. But there's a few things that I want you to note. Like, for example, if you look at item C, you'll see that the expected ratio is 10 and the mass spec version is 0.3. So there's a terrible discrepancy between what we actually know to be true from a structural biology point of view and what the measurements tell us. The other thing I would say is just that the reported accuracy of these measurements, or I should say the reported precision, is stunning, and well, actually, I don't think it's true. They're reporting things down to single digits, and I guess I just wanted to say that, one of the motifs of what I'll be saying in this course that I'll come back to again and again is, I have strong negative feelings about the current state of surprise, quote unquote, in our current science, in the way that people write referee reports and so on. And frankly, I believe in this day and age, especially in this day and age of fake news and alternative facts and all that other nonsense, that in fact what we want to celebrate is unsurprise. We want to celebrate when different methods tell us exactly the same story. There's no surprises. We know how to measure a thing. We don't report numbers with some ridiculous lack of understanding of precision, and we get reproducible results. So this is 15 different methods of measuring Avogadro's number. It's from the book Atoms by Jean Perrin from more than 100 years ago. And I simply wanted to point out that, it's, first of all, the ingenuity of the different ways of measuring Avogadro's number is super impressive. And then it gives us a sense about how well we know the number, because these different methods, they don't agree perfectly. Okay, question. The genome of Vibrio cholera is 4.03 megabases. How many genes does it contain? So A, less than 3,600, B, 3,600 to 4,400, and C, greater than 4,400. So the way that I would think to reason this out is I would say, well, let me comment on the typical protein sizes. So typical protein sizes are, let's say, for more or less all organisms, I'm going to say few times 10 to the 2. So I'm going to commonly use this notion of few, which means on the order of 3, but it really means few. The arithmetic that I will adopt, as suggested by Sanjoy and Mahajan, is few times few is 10, and 1 over few is few over 10. So those are the key rules. So at any rate, typical protein is few times 10 to the 2. in amino acids, that means on the order of 1,000 nucleotides will need to be used to code for that protein. And so if the genome is strictly protein coding, then I take the size of the genome, divide by 1,000, that tells me how many genes. And so this is an example of that postulate. The data shown on the right from Mike Lynch's book on the origins of genome architecture is really cool because it basically tells us that the number of genes scales linearly with the size of the genome, and the slope is 1 gene per 1,000 base pairs. That's a very nice estimate. This one I am going to skip. We'll talk about it later. So the summary of the first part of our little exam is just to tell you, first of all, that I hope that you'll consult the BioNumbers database. This is something that was produced primarily by Ron Milo and Uri Moran. And this database is meant to codify and to vet the key numbers about biological systems in a way that you ought to be able to trust. And the thing I would say is that, what we would like to do is have sort of a database of the relevant numerical quantities for biological systems that rivals what we have now, for example, in the physics and chemistry handbook, which tells me whether it's the specific heat or the thermal conductivity or the electrical conductivity or the fracture toughness or whatever. Those are reliable numbers that we can use when we try to think about the material world and design things. And I think that one could ask for that same kind of level of accuracy in the context of these bionumbers. So with that, I'm going to wrap up this exam. I have a second part, but I think I'll hold that second part for later in the course, having to do with human impacts.